Okay, so hi. Uh, I hope I'm audible. Yeah. So, good evening and welcome to today's class. Today we'll be beginning week 15 for the course and taking just a second. Yeah. So, taking us back to the discussion that we had in the previous session, there was a bit of a disconnect, so I'll just quickly brush up the topic. So, if you remember, we had completed a large part of the course already. We only have a few more classes left, and we'll cover the remaining topics in that duration. So, we had already completed. Oh, just a second, I accidentally put this slide. I need this one. So, yeah. So, in the previous class, we had begun our discussion on understanding DNA protein interactions because we had begun our discussion on DNA protein interactions through because we wanted to shift our discussion to DNA protein interactions using high throughput methods. And in this context, we wanted to initially start a discussion with core techniques which are necessary to study DNA protein interactions. And that will be the focus of our discussion for today. But before we move into the techniques, we had initially begun a discussion on DNA protein interactions. What are the different types of proteins that can potentially interact with DNA? What sort of interactions stabilize these, like what sort of bonds or what sort of forces stabilize these DNA protein interactions? What should be the nature of this interaction? How stable it should be, or whether or not at all it should be stable? All of these different factors have to be taken into consideration. So, with this introduction in mind, we had started the discussion in our previous class, where first of all we understood that the way in which a DNA molecule and a protein molecule interact with each other is actually something of a lot of biological significance. Why do we say so? That is because if you think about any process which is regulated within the cell, you will find that more often than not, it is a protein that is controlling that regulatory process. When I say regulation, it can be something as simple as think about a restriction digestion for that matter, something that you and I in the laboratory will perform without a second thought. But if you think about the biology that goes behind that execution, that is quite intense, that is quite extensive, and it is regulated, of course, by proteins. What proteins are these? These are proteins that are functioning as molecular scissors. They're able to recognize a sequence, bind to it, and then carry out an enzymatic cleavage at that site within that sequence. So, with this introduction in mind, we should keep it. We should also have perspective regarding this fact that it is not only a protein which can execute any information or which can carry out any process just by itself. It is a mediator, it's a regulator, and it's a controller, but a protein has to interact with A, other proteins, B, with nucleic acids, and in different contexts. And particularly in our case, currently our focus is on protein and DNA interactions. A large portion of this DNA protein interactions are usually carried out by transcription factors. What do we mean by those? Transcription factors are those proteins which are able to bind to DNA at some specific consensus motifs or some recognition sequences, and then they are able to carry out or regulate or control transcription from that particular site. They're able to upregulate transcription from that sequence, and then they're able to synthesize more and more of that corresponding genes RNA, which can then undergo translation and then synthesize that protein. So, because of the crucial nature of this interaction and uh, the overall cellular control that transcription has, because we already have this idea and it's beyond not even beyond, it's actually primitive to the scope of this course, that when we talk about transcription and the essentiality of it, we understand how crucial it is to control it. It has to be sensitive to any stimuli that might instruct the cell to shoot it up or bring it down based on whatever the conditions the cell is able to sense at that point of time. And secondly, also, transcription needs to be under the control of not one protein, but instead under the control of an entire network of proteins, so that any fluctuation or any alteration in one particular protein sequence does not completely disrupt a transcriptional network. And there are multiple checkpoints that can be put in place before anything goes awry. So, with this introduction, we'll move forward. And we had begun our discussion in the previous class talking about the fact that protein families, uh, most of which are all eventually going to function as transcription factors, can bind to DNA in different ways, depending upon what sort of complex is formed. So, the majority of these interactions, they have a number of facets that are usually considered. We didn't go into too much detail about it, but that includes the protein's size, the DNA binding site, the strength of the binding, the effect of the protein binding to the DNA structure, what bases are involved in that interaction. All of these factors are to be considered. And on the right, what you see is a representative image, a schematic of a DNA protein interaction, which has been modeled using any common software like Chimera or PyMol, where it can simulate what the DNA looks like and how the protein can interact with it. And this was a ribbon diagram that represents the structure of the protein. And if you zoom in, you can exactly figure out which bases are there. You can find out which forces or which interactions are stabilizing this DNA protein hybrid. So not hybrid, but DNA protein interaction. So all of that information can be figured out. Now, like I already mentioned, whether complex or simple, a major function of these DNA protein interactions is to work as genetic switches. They're able to control the expression of a gene and the relevance of a gene within the cell at any particular point of time. So proteins are able to recognize these sequences and carry out the transcription of these genes. And typically these cis-regulatory elements to which these transcription factors actually bind, they lie pretty close to the gene, often in the intergenic region, which is directly upstream of the transcription start point of that gene. And therefore the position, the identity of the gene, the information which is embedded in that genome, all of that ultimately determines the time and the place at which each gene gets transcribed. Now, how do these different base pairs of DNA get recognized without the need to open up the DNA double helix? So there are four possible configurations. I don't think I have placed it here. Yeah, I did. So there are four possible configurations in which these interactions can occur. We had talked about the fact that transcription factors, most commonly, they bind to the major groove of DNA. I think I had, yeah, maybe this, or I think. Anyway, basically the major groove gives sufficient area for the transcription factor to bind and to find its recognition sequence and to form sufficient bonds to stabilize that interaction. So major groove is the site of recognition. And yeah, apart from that, if you look at the double helix, the outside of the double helix is studded with DNA sequence information, which the transcription factor or regulator is able to recognize. And the edge of the base pair, for example, these base pairs, the edges of these, they present a distinctive pattern of hydrogen bond donors, acceptors, and hydrophobic patches in both the majors and minor grooves. But among them, because the major groove is wider and it has more molecular features than the minor groove, nearly all transcriptional regulators make most of their contacts with the major groove itself. Now, the binding of this transcriptional regulator to a specific DNA sequence can be seen, where you can see that some single contact between a transcriptional regulator and DNA is facilitated, which allows the protein to read that DNA sequence. And a complete set of contacts can also be often seen. So I think this image was here. Yeah, so then we moved on to our discussion on DNA binding proteins. There are helix turn helix proteins. Okay, there were four major classes that we discussed: helix turn helix, helix loop helix, beta sheet recognition proteins, and zinc finger proteins, which are a major class of the same. So if you look here, helix turn helix proteins. Originally, they were identified in bacteria, and this motif has been found in hundreds of DNA binding proteins from both eukaryotes and prokaryotes. It is constructed from two helices, hence the name. Shown here in red and in blue, which is connected by a short extended chain of amino acids. So you have an amino acid stretch as well, which is called as a turn. Now the two helices are held at like a fixed angle, primarily through interactions between the two helices. The more C-terminal helix is called the recognition helix. The red one in this case is the recognition helix because that is what binds to the DNA major groove, and its amino acid side chains actually facilitate the interaction between the recognition helix and the DNA double helix. And all of the proteins shown here, they bind DNA as dimers, in which the two copies of the recognition helix are separated by exactly one turn of the DNA. Therefore, both the recognition helices of the dimer can fit into the major groups. Mathematically also, that has been justified. Next, soon after, genetic analysis of the Drosophila fruit fly led to the characterization of another important class of genes called as the homeotic selector genes, which play a critical part in fly development. And it was shown that these genes, they coded for transcriptional regulators that bound DNA through a structural motif called as the homeodomain. So this homeodomain is an entirely large group of proteins also which function as transcription factors, not just in Drosophila but in eukaryotes as well. And two different views of the same structure have been shown. First, you can see the three different helices. Again, the red one is the recognition helix, which is facilitating direct interaction with the DNA, and then you have helix 1 and 2 as well. And the part that contains these helix 2 and 3, the recognition helix and the other one, they resemble the helix turn helix motif. Now, the recognition helix 3, shown in red, forms important contacts with the DNA groove. And one example of the same is the asparagine, which contacts adenine of DNA. As you can see, these are multiple interactions that can also be facilitated. And a flexible arm that is attached to helix 1 that forms contacts with nucleotide pairs present in the minor groove. So you have a two-pronged approach. One approach by the protein, by the recognition helix, with the DNA backbone, with the adenine present in the DNA, and the other helix has an arm which is able to form interactions with the nucleotide pair that is present within the minor groove. So this was as an example. Then among the excuse me, among the different motifs, the leucine zipper motif is named because of the way that these two alpha helices, one from each monomer, are joined together to form a short coil structure. So these proteins, they bind as dimers, where the two long alpha helices held together by interactions between hydrophobic amino acid side chains. Usually these are leucines that are present at the dimerization interface between the two coil structures that you can see. And just beyond the interaction or dimerization interface, these two helices are separate from each other and they form this sort of pie-shaped structure, which allows the side chains to contact the major groove. So you have like a set of leucines, polyleucine sort of stretch that is present, that is allowing these two to dimerize with each other. And then beyond that, you have multiple interactions stabilizing the protein with the DNA interacting with each other. The dimer therefore sort of grips it like a um, sort of like a clothespin, like a clip that you have on a clothesline or on a rope. So that is what it looks like. And another reason for the name is much like the rungs of any zip are being resembled here by the leucine residue. So you have two leucines which are interacting with each other, much like a zip runner. So hence giving rise to the name, the leucine zipper motif present in proteins. Then you have beta sheet DNA recognition proteins, which are another set. These are a little unusual. More commonly, you will find alpha helix based motifs to be more prevalent for DNA recognition. However, there's one group of transcriptional regulators which form or which assume a two-stranded beta sheet structure with amino acid side chains extending from the sheet towards the DNA, reading the information which is present on the major groove surface. Now, this beta sheet motif also is capable of recognizing multiple different DNA sequences. The exact DNA sequence recognized depends upon what amino acids are actually composing are part of that particular beta sheet. So this is the transcriptional regulator, which binds to molecules of SAM or adenosine methionine. Here you can see it in red. And on the left is a dimer of the protein. On the right is a simplified diagram, which shows how the two-stranded beta sheet binds to the major group. So it sort of binds flat on the DNA molecule and has multiple recognition sites for the same. Next, you have helix loop helix proteins. So much related to the leucine zipper. The helix loop helix motif consists of a shorter alpha helix connected by a loop to the second longer alpha helix. The flexibility of this loop sort of allows one helix to kind of fold back and park against the other, forming the dimerization interface. So it is similar to what we have seen for leucine zipper, but there the dimerization interface is a lot tighter and it's mediated by the amino acids which are forming that interface, versus for the HLH or the helix loop helix motif, it's different. So as shown, this two helix structure binds both to DNA and to the two helix structure of the second protein to create a homodimer or a heterodimer. Here, the representation is that of a heterodimer. Two alpha helices that extend from the dimerization interface make specific contacts with the major groove of DNA. Then we also have zinc finger proteins, which are ZFNs, which this is something we spent a lot of time on. This group of DNA binding motifs includes one or more zinc atoms as a major structural component, and all such zinc coordinated DNA binding motifs are called as zinc fingers. So the reason it sort of resembles the structure of or uh, the structure resembles something like that of a finger, at the center of which you have a zinc atom, which is forming multiple coordination bonds with C and H residues, which C and H amino acids, cysteine and histidine amino acids. And that sort of this coordination complex stabilizes this entire structure. And these ZFNs are a major group of transcription factors as well. And the zinc atom holds an alpha helix and one beta sheet together, for example, in the middle. This is often found in clusters with the alpha helix of each finger. So you have multiple such motifs present, and the alpha helix of each finger contacts the major groove of DNA, forming a nearly continuous stretch of helices along that groove, and that builds up a strong and specific DNA protein interaction. And that's what you see. Here you see three tandem zinc fingers present, blue, red, and green, which are interacting with the DNA. So that is what we saw. And then we talked about transcriptional regulators. So a monomer of a typical transcriptional regulator recognizes around 6 to 8 base pairs or nucleotide pairs of DNA. However, sequence-specific proteins do not bind tightly to a single DNA sequence and reject all others. Rather, they recognize a range of closely related sequences with the affinity for the protein for the DNA. Um, so how closely, so this is actually a representation of any DNA motif which is recognized by a sequence. In this case, it is for NANOC. So it is a homeodomain family member, functions as a crucial regulator in embryonic stem cells. And often you'll find motifs like this using tools like JASPAR, which is a database where you can based on all the studies that people have already carried out based on literature survey, you can sort of screen for motifs. So the logic of it is, for example, if you want to say that a gene X is regulated by a protein Y, then if you have the binding sequence or the recognition motif of Y, you will scan for that motif on the upstream promoter or promoter upstream region of gene X. And if you find that motif to be present, that is one putative site for binding of your gene of interest. The other possible ways of strategizing the same thing is by looking for ChIP-seq data, and we will talk about it as well eventually when we discuss in vivo DNA-protein interaction techniques. Today we'll be discussing in vitro. So yeah, that is how transcriptional regulators bind. And then we talked about how what are the ways in which this binding can occur? Either it can be direct binding or it can be cooperative in nature, and which basically means more the amount of protein which is available for binding, more DNA occupancy is observed, and you see this sort of sigmoidal plot, which represents cooperative binding. And then we also talked about the fact that in general, the binding of transcriptional regulators to DNA occurs because of some chromatin remodelers assisting, because they open up the heterochromatinized or the nucleosomal DNA, and they make it more accessible, and they allow the recruitment of these proteins, these remodelers, which holds the DNA in that naked conformation, so that accessibility is now feasible for all of these protein regulators. Otherwise, DNA would never at all get transcribed. Apart from that, we also talked about nucleosomal breathing, which is basically a phenomenon whereby very short regions just beyond the nucleosomal DNA, sometimes often even considered to be within the region where histone H1 protein binds. That region is often accessible to transcription factors because they are able to find a patch of nucleosomal DNA, previously nucleosomal DNA, to which they can access. So yeah, that was pretty much the discussion we had in the last class. Now, today our discussion is going to shift towards in vitro DNA protein interactions and what strategies can be used to study the same. Now, before getting into the detail, one very crucial, very fundamental thing for us to understand is that DNA protein interactions, what do they mean? They mean simple, like what do I mean by this question also is that if you simply have one piece of DNA and one protein, do they interact with each other directly? When you do such an investigation, when you checked the possibility of two molecules interacting with each other outside of their nascent surroundings, or you are doing it physically, you are directly checking for the binding of a protein onto a DNA sequence. One strategy to do that is using these in vitro methods. But when your objective is to see what happens within the cell, within the cellular environment, under a particular condition, plus minus a stimulus, plus minus some drug, or whatever XYZ condition you're interested in, when you are checking for a DNA protein interaction under those circumstances, then that is an in vivo method of study. Now, today we'll not be discussing in vivo method, but you can actually, that will actually be more relevant in a biological scenario. But when you are doing more of a structural analysis and you simply want to check for binding of protein X and gene Y, then you can opt for these in vitro strategies. So that was the overall introduction. But because of today's class being a while away, I wanted to incorporate a bit of an intro to the discussion. However, I've already spoken a lot about it, but let's go over it anyway. So, association of DNA with proteins is a phenomenon of utmost importance. In effect, almost all aspects of cellular function, such as transcriptional regulation, chromosomal maintenance, replication, DNA repair, all of these depend on the interaction of proteins with DNA. Activation of genes by DNA binding proteins. Yeah. Activation of genes by DNA binding proteins is a fundamental regulatory mechanism. We've already discussed that. Which involves chromatin remodelers, transcription complexes, all of which will assist in the initiation and the elongation and the continuation of gene transcription or RNA synthesis. Such DNA binding proteins, they have a number of diverse roles and might function as structural proteins that make up the nucleosome, enzymes modulating chromatin structure to control gene expression, transcription factors, sometimes also as cofactors. One of the most widely studied examples of DNA binding proteins is a transcription factor, which has been our focus in the previous class as well. It is considered to be extremely crucial, this interaction, in developmental processes and, of course, also in response to environmental stresses. Also, in humans, their dysfunction can contribute to the progression of various diseases. And in lieu or in view of such an important role, various techniques have evolved over the years to elucidate them. And each technique, please bear in mind, depends on depending on what your objective is, what question you wish to address, the pros and cons, and the accuracy of the technique you're using will very largely depend upon what question you wish to ask. So each of them serves a very specific purpose, and most commonly, these techniques can cater to either of the two parts of the interaction, meaning that either the technique can focus on the protein of interest, which means the protein's molecular weight, its identity, its domain, etc., or the focus might be on the DNA, the general sequence, the specific sequence, alternative sequences, etc. So here, what you see on the right is this just as a reference of a very beautiful structure that was decoded at Berkeley Lab by Susan Sutakawa. So here you have the structure of FEN1, which is an endonuclease, and how it binds to its target DNA. Again, this is an example I already gave because we had discussed restriction enzymes also in the previous class, and it's something you can very easily sort of visualize. So researchers here found that this is a bit of an old paper, I think in 2017 or 18, a couple of years ago. But researchers found that single-stranded flaps get threaded through a tunnel present in FEN1, and the unexpected inversion of the threaded flap, guided by phosphate steering, keeps the phosphodiester bonds facing away from the metals that could inadvertently shred them. So this is how the DNA protein interaction is facilitated in this case. In vitro techniques to study DNA protein interactions is going to be our focus. So where do we begin? First assay, we'll not be discussing all of the assays because that is going to take forever, but we're going to discuss a couple of them in a lot of detail, and a few I'll just introduce you to so that you can read about it in your own good time. Uh, The first one we talk about is a footprinting assay. So what is the logic of it? The logic is that any protein which binds to DNA can leave behind a footprint by virtue of its DNA binding capacity. So footprinting assays are based on the principle of protection of protein-bound DNA from degradation. So any region of the DNA which is bound by a protein is protected and remains undigested while the rest of the backbone becomes completely chopped up. The technique is used to decipher a specific sequence to which a DNA binding protein or a molecule binds. The procedure employs chemical or enzymatic digestion of naked and protein-bound DNA oligomers. Both these reactions are then compared using gel electrophoresis, and the segment of DNA which is bound by the protein sort of appears as an empty patch. Just for a quick reference, you can see here on the right, this is a region which has not gotten digested when compared to the continuous fragments which are produced by naked DNA digestion. So let's go with the procedure first. Here is your DNA, which is labeled at the 3' end. Uh, why at the 3' end? Why not at the 5' end? Well, you could go either way, but it's a good practice to opt for the 3' end because that will keep a continuity for all of your markers, and you'll get a range or a smear of bands of different um, molecular weights of different kilobase sizes, so that you can actually visualize where which patch is actually lacking. So this is your set of labeled DNA to which you either do not add protein or you do add protein, and then you carry out partial digestion by DNase 1. We'll go into partial digestion in a couple of slides, but basically what DNase 1 does, as the name naturally indicates, it's also an endonuclease. It is going to cleave DNA. That's what it does. So any region which is not sterically hindered or is not interrupted by a protein or not protected by a protein is accessible to DNase 1 and is capable of undergoing digestion. So that is what happens. And on the left, you can see when there is no protein or protection, complete DNA digestion is observed, and this is the expected sort of um, a result of this chopped up DNA. You run it on an agarose gel, and you can see that there's a range of molecular weights to which these fragments can belong. However, when there is protection by or protection and steric hindrance presented by a DNA which is bound to some protein X, then in that case, that region remains sort of thin sealed and inaccessible to the digestive enzyme, and as a result, that region remains sort of undetected and pops up as a footprint of the protein binding to that DNA. So that's what you see here. And on the right, you can see this is your sequencing ladder, which shows you the molecular weights which you expect. And on the extreme right, you have a control reaction, which is um, which is a known footprint, which does not have the expected footprint. Then among the footprinting reaction, you have plus and minus protein, and then you get your outputs. And what you see on the bottom is a representation of such a result, where you can see that in the fourth lane there is complete protection, and there are no fragments of this particular size, and as a result, you do not see any fragments in this range, and so they are all protected. So again, something extremely cool and extremely nice to study. And this represents the actual sequence which is present within that region, which is bound by your protein of interest. Following that, footprinting has been essentially been used as a tool for elucidating sequence specificity and the dissociation constant of a variety of ligands binding to DNA. The agent which is used to cleave DNA is called the probe. So the smaller the probe, the higher is the resolution provided, but its chance for cleaving the DNA under the bound protein also increases. The enzymatic digestion method, it involves the use of either DNase 1, like we already discussed, then MNase. I think we have discussed MNase as well, the way in which micrococcal nucleus cuts. Then you have MPE, which is methidium propyl EDTA free, EDTA Fe2, chelated iron. And then you have copper, phenanthroline, uranyl photocleavage, hydroxyl radicals, iron complexes, etc. All of these different strategies can provide you various sorts of information which you can use based on what your research question actually is. Now, among all the possible footprinting assays, DNase 1 digestion is the most commonly utilized. It is a double-stranded specific endonuclease which sort of binds to the minor groove, and its job is to break a phosphodiester bond. The technique was developed by Gallus and Smith for visualizing the binding of the lac repressor protein to the lac operator sequence. So the footprinting technique employs the use of single-end radio-labeled synthetic or natural DNA fragments. This fragment you can again, so basically what you're doing is you have a single end labeled DNA, like we already mentioned, the 3' end, the 5' end is usually the preferred end of labeling. I cannot remember what I said previously, but yeah, the 5' end, the left end or the initial synthesis end is preferably always labeled to make it easier to detect. And then the fragment is incubated with either crude or purified protein sample. So preferably if you wish to um, study the binding sort of nature or the binding of a particular protein, then it is usually preferable to go for a pure protein extract. And under appropriate binding conditions, you would allow your protein to bind to the specific DNA. And this protein bound fragment and the control naked fragment can then be subjected to DNA spun treatment in an appropriate buffer with varying concentrations, time periods, etc. Then you run it on a DNA screening page, you process it, and you image it, and you get your results. Now, this is a representation of how DNS1 hypersensitivity can also vary. So you have a region of exposed chromatin, or rather, a better way to put it is accessible chromatin. And this is a nucleosome free region, hence the capacity of DNase1 to actually bind there. And apart from that, you might, that is actually the enhancer. So another readout of a DNase1 footprinting assay, usually if you carry this out with say for example a known transcriptional regulator like RNA polymerase 2, or rather serine 2 phosphorylated RNA polymerase 2, then you check for its check for the different locations of binding. Then what you get as an output in that case will indicate sites of active transcription and transcription, or sites of active expression within the cell under those conditions. So that can either be in the enhancer region or in the promoter region, nucleosome free promoter region, or there might be nucleosome sort of repositioning in region A or region B, and then transcription goes up in these sites also. The protein bound fragment and the control naked DNA fragment will be subjected to DNase1 digestion, and then after you run it on the gel, you will be able to visualize the pattern of the footprint. Uh, there are a couple of considerations to be kept in mind whenever you do decide to perform this particular technique of DNase1 footprinting. First, by altering chemical conditions or experimental conditions, DNase is partially allowed to digest the fragment, assuming that there has only been one single nick per fragment. Why is this necessary? If you add too much of an enzyme, then beyond a threshold, non-specific digestion is actually possible. So therefore, it is definitely a good practice to dilute your conditions such that DNase makes only one cut per molecule, maintaining the procedure. So this creates, as a result, a range of fragments which got distracted. Just give me a moment. Yeah. So, upon examining a variety of experimental footprinting gels, it is evident that if all the sequences are cleaved without any sequence-dependent specificity by DNase1, then all the fragments would technically have been of the same intensity. But because DNase1 has partial sequence specificity, resulting in some sites becoming hypersensitive, and as a result, what you see in those sites is a more intense band compared to others. So, yeah, next, end labeling of DNA is also extremely crucial. Why? In one reaction, DNase is going to cut both strands. That leads to a mixture of positive and negative strand fragments, which are further separated on the denaturing gel. Yeah. The purpose of radio labeling DNA basically, or in any way labeling DNA either by a fluorescent tag or by some other moiety, which is going to make it visualizable and detectable. So the purpose of this DNA is to provide a clue as to which of the two antiparallel strands does the protein under consideration bind. So basically, if you label the 5' end your uh, of your DNA of interest, then you would be able to figure out that which end it is actually binding. If it was binding to the other end, then you would find the motif to be present on the antisense strand. and not only side strand, and that can actually help you in segregating both of these conclusions with relatively decent confidence. So it is common to digest both the positive and the negative strands in separate tubes and then run them alongside each other. Lastly, there are a variety of methods to analyze the final footprinting image, which range from either visual inspection to creating a differential cleavage plot based on densitometric analysis. There are other techniques also which are available for quantitative analysis of binding affinity. Now, what we had initially begun our discussion on was on the role of DNA's one digestion, and not just simply digestion, rather partial digestion having a major role to play. If there was complete digestion, it would have chopped up, your entire nucleic acid population, and that, of course, is not something which is desirable. But instead, you want to um, sort of control its activity such that it can do so. It can only generate one cut per molecule, ensuring that there is some specificity which can be maintained. So bovine pancreatic deoxyribonuclease, or DNase-1, which is a common source for DNase-1, is an endonuclease. This endonuclease is capable of cleaving both ssDNA and dsDNA to produce primarily 5' dinucleotides and 5' phosphate dinucleotides, 5' phosphate oligonucleotides. So DNase-1 degrades dsDNA or double-stranded DNA, however, in a completely non-specific manner. The enzyme can utilize either of these two cofactors, either Mg2+, which is magnesium, or it can utilize Mn2+, which is manganese, for full double-strand cutting activity. In the presence of only Mg2 plus alone as a cofactor, DNase-1 shows its partial digestion effect. And instead of, generating double-stranded cuts and chopping up DNA, instead it exhibits a nicking activity, and the single-strand nucleotide activity is much more discriminatory. So that is overall what we see here. So after you have done this, so if you remember, we had first used DNAs for partial digestion of our sequence of interest. Second, we justify the logic of end labeling of DNA. Third, you have to run a denaturing SGS page. Why? Because you do not want any heterodimers to be present in your basically, you do not want double-stranded DNA, or you do not want anything which falsifies your results. And so for that purpose, running a denaturing page is going to make sure that only the radio-labeled probe containing DNA is recognized, and often the naked DNA is chemically sequenced and run on the same gel as a marker for finding the exact sequence of the footprint. However, some precautions need to be considered. Uh, what are these precautions? First, it is important that you do not titrate too much DNA with the protein sample. Why? Because it causes a large amount of DNA to remain unbound from the protein and thus susceptible to DNase-1 attack. This fragmented DNA will show up at the place of the usual footprint, making it much, much harder to detect. Second, the incubation of crude samples with the fragment requires the presence of competitor DNA. Why do you need competitor DNA? Because that will help in exclusion of any non-specific binding proteins from coming up as a footprint. The limitation, however, of this technique is that it does not provide identity of the protein. It only tells you that an interaction um, exists and is necessary. Because of the large molecular weight of the DNase-1, its attack gets sterically hindered by the bound protein. Other probes for nicking DNA, like free radicals, may not be hindered so easily and hence have a chance of nicking a few bases under the bound protein itself. Now, this is what we are saying are the two precautions to be taken care of. One, that you don't titrate too much DNA, which will all remain unbound and that is going to get digested by DNase 1. And second, the incubation of crude samples with the fragment requires competitor DNA to be present so that all non-specific DNA can be excluded by showing up as a footprint. Yeah. So get the protein's identity. However, because of the large molecular weight of DNA is 1, its attack gets hindered directly and by the bound protein and other probes for nicking DNA like free radicals might not be hindered so easily and hence might have a chance of nicking a few bases under the bound protein itself. Now, apart from what we have just discussed, the footprinting method is also used for other purposes, like for example, for RNA's footprinting, for any other utilization as well. It may any other protocol also it might be used. For example, often it is used in DNA's seq, where you have DNA's one digestion followed by sequencing, used for genome-wide studies and protocols using automated infrared sequencers, allowing long-range and highly sensitive DNA's one footprinting have been developed. Yeah. So DNA-seq is often used for genome-wide studies. Now, protocols that use automated infrared sequencers allow for very long-range sequencing. If you remember, we had discussed long-read sequencing before, and also very highly sensitive DNase one footprinting is also performed. Also using streptavidin-bound oligonucleotides for protein binding and subsequent DNase one digestion makes the technique very convenient. It can also be used to fish out a protein of interest from a crude sample. It is also used for quantitatively measuring the dissociation constant of the protein DNA interaction. So this is one method. The second technique that I wanted to discuss today is called as EMSA or electrophoretic mobility shift assay. Again, this is not a very recent technique, but it's very informative, very useful. It is a relatively simple in vitro technique to study DNA protein interactions, and its novelty lies mainly in its application to deduce the binding parameters and relative affinities of a protein for one or more DNA sites or for detecting basically protein nucleic acid interactions, whereby you wish to compare the affinity of different proteins for the same cell. It is simply based on the principle that DNA protein complexes are heavier and they move slowly when subjected to non-denaturing polyacrylamide or agarose gel electrophoresis as compared to the unbound free row. Yeah. Since the rate of DNA migration is shifted or retarded when bound to protein, the assay is also referred to as a gel shift or gel retardation assay. The DNA sequence is provided externally and you can incubate it with the crude cell protein lysate. Yeah. So since DNA migration rate is shifted or it gets retarded, you will be able to figure out that there is an interaction between the two. So here you have a representation of a tube, which is basically confirming that this is an in vitro method. You are separately adding your cell lysate and you're adding your oligonucleotide of interest and oligos and proteins from the cell lysate are incubated together to allow the formation of your DNA protein complex. And DNA protein complex can also be incubated with antibodies, which are specific to the protein of interest to see a super shift. So then you can run it on a gel and you can visualize the same. We will look at better representation of this. In fact, we will look at an example for the same for better understanding. So this is an example of an ENSA experiment. So to perform the assay, a fragment of DNA which carries the suspected binding site or the suspected interaction site is labeled by the addition of 32 phosphate, which is a heavy isotope of phosphorus, using the T7, yeah, using the T7 DNA kinase, which is going to allow the introduction of this 32P. And now, if the transcription factor that will bind to that particular site or to that particular region is present, then you'll be able to visualize a new band by autoradiography that will correspond to the protein DNA complex. In this study, the example taken was of ERF1 AP2 gamma, which was first identified by gel shift analysis using the ER promoter regulatory element. Yeah. So you have your sequence of interest. This is the ER coding region. This is the activity site. You take the DNA from this region, you add your protein, the bound complex is then run on a gel. You see, this is the free probe and this is your bound sequence. So this is the concept behind it. Now, practically, if you do perform this experiment, if you do perform EMSA and you do run it on the gel and you do show it, what do you expect to see? So here you have MCF7 cells and the MDMB231, which are breast cancer cells. And in a breast cancer cell where there is no competitor, nothing else present, what you see yeah, when you have your wild type 13-mer sequence to which the nascent protein is able to bind, the ERF1 AP2 gamma, this particular protein, when it is able to bind, you will find this particular sequence to be present. Now, yeah, when you are adding competitive DNA to your cell lysate mixture, what sort of proteins are you going to see? This is your expected sequence, which is the palindrome that is required, which is the wild type sequence, which competes with the binding of the DNA. Now, this wild type DNA is not labeled, and as a result, when no competition is present, you get this distinctive band to be present. Versus when you are adding this competitive DNA, you can see that there is no band which is present. Now, when a range of different mutants are taken up, where two bases at a time are mutated within this particular DNA sequence, and then the loss of binding is observed with the addition of this competitor, then what you see is that, yeah, when you look at these three or four mutants, you can see that there is partial blockage or partial competition by these mutant sequences. However, mutant 13, 14, and 15, as well as mutant 1 and 2, you see that there is a complete loss of this particular interaction. And as a result, that implies uh, that this central region is crucial for the binding and therefore needs to be taken further for study. In addition to the standard protocol that we explained, the supershift assay is performed to specifically assert that the DNA protein interactions that you are seeing between the two entities by using an antibody which is specific to your protein of interest. So under these circumstances, by incubating the antibody along with your DNA protein sample, followed by running it on a gel or gel separation, the DNA protein antibody complex may be visualized as a supershift band. Competition assays may also be performed using unlabeled specific and non-specific oligoduplexes. Now, EMSA may be used qualitatively to identify sequence-specific DNA binding proteins in crude lysates and in conjunction with mutagenesis to identify the important binding sequences within a given gene within a given gene upstream regulatory region. So what is your objective? Your objective is A, detect the possibility of a DNA protein interaction, B, to qualitatively study it, C to check for the sequence specificity of that interaction, which is what we saw in the previous slide, that if you alter a couple of bases, one or two bases at a time within the recognition site or recognition motif, how does it affect this binding capacity that we've been talking about? So EMSA can also be used quantitatively to measure thermodynamic and kinetic parameters. So this technique, it poses several advantages. The most significant benefit of EMSA is its ability to resolve complexes of different stoichiometry or different conformation. Another one is that the source of the DNA binding protein, either you can have pure complete protein or you can have some sort of a crude nuclear extract, in vitro transcription product, or a purified preparation. In addition, the relatively low ionic strength of the electrophoresis buffer helps to stabilize transient interactions, permitting even labile complexes to be resolved and analyzed by this method. So that is what we saw here. Yeah. Then you have another assay. I think that's the last one. Yeah. Just give me a moment. Oh yeah, that is our last slide for today. Yeah. So the third technique that I was talking about involves proximity ligation assay. So what is that? As the name suggests, it involves the proximity between two interacting proteins, and it is a I mean, it's a different strategy that works a little differently compared to what we have talked about, but it is still included under in vitro techniques. Hence, I thought I would bring it up. And secondly, also because this particular strategy is a little bit costlier, but it is relatively new and is a short confirmation of the interaction that we are talking about. So it is considered to be an ultra-sensitive protein analysis for measuring DNA-protein interaction. So in this technique, the direct detection of proteins or DNA-protein interactions is possible, and DNA representations of detected proteins are created. So following this, the amplified oligonucleotides get attached to specific protein binding reagents like monoclonal or polyclonal antibodies. And one of the proximity probes that we talked about is a partly double-stranded oligonucleotide with single-stranded extension. The other probe is an antibody directed against the DNA binding protein, and it has an attached DNA strand with a free 5' end. So what do you see here? You have these two targets, which are, say, for example, also binding to DNA. I think I could have chosen a better image for it, but okay, let's see. So these are two primary antibodies which are binding to your protein of interest 1, protein of interest 2, and the primary antibodies bind to the targets when primary antibodies detection with species specific secondary antibodies and PLA probes which are conjugated to oligonucleotides. So these are your PLA probes and these are your antibodies. Then hybridization and ligation of these two, they like basically to put it straightforward, when you have two probes which are in very close proximity of each other and they are able to bind to the same target molecule, or they are able to they are located very close on DNA. It's actually a better method of protein-protein interaction study, but it can also be used for DNA-protein interaction as well. So the two target interacting molecules, when they're very close to each other, then the ends of their conjugated oligonucleotides get joined by enzymatic ligation by the addition of a connector oligo, and the detected protein molecules will promote ligation reaction by ensuring that there is sufficient proximity between the two. So after hybridization and after ligation by the ligase, as you can see here, rolling circle mode of amplification is taken up and sorry, excuse me, and detection by labeled complementary oligonucleotide probes is also carried out. And this method is highly specific, very sensitive for solution phase analysis of interactions. Large sets of proteins can be actually analyzed in parallel. And this assay can also be a very valuable tool to characterize sequence specificity of DNA binding proteins and to evaluate the effect of polymorphism in transcription factor binding sites or TMPS. So that sort of ends our class for today. But because we have some time on hand, I am going to quickly go over the topics, a couple of topics that I have not covered today. Uh, just give me a moment. So, in today's class, this is the book and the reference that I've gone through. Oh, let me just share my screen. Yeah. So this is the chapter that I've referred to. You can go over any textbook that you are interested in, but I particularly wanted to bring forward this because it's a very composite way of showing it. So here you'll see in vitro techniques for DNA protein interaction from where I showed you DNA footprinting assay and electrophoretic mobility shift assay. But there are also other methods which I didn't get into. But there is southwestern blotting as well, which is southern blotting with combined with Western blotting. Let me just go to the full size image for your reference. So here you can see you have your purified or your fluid cell extract and you run it in a 2D or 1D SDS page. Then you add labeled probes and you can detect your protein complexes. Then you have yeast one hybrid assay. There's also yeast two hybrid assay, which is for DNA, which is for protein-protein interactions of PPI. But DNA protein interactions are different. This is a positive interaction between a DNA binding protein and the bait. And there's a reporter gene which is present downstream of it, that whenever there's an interaction of these proteins, then DNA protein interaction is getting validated and you're getting a readout of your luciferase reporter. And when there is no interaction between DBP and the bait sequence, then no reporter driven action can be taken up. Then you have the yeast one hybrid, that is the yeast one hybrid assay. In other ways, you can also refer to as a reporter assay for understanding as well. Then you have the fudge display assay, where you have biogenylated oligonucleotide sequences, which are incubated with magnetic beads or agarose beads, which have been coated with streptavidin. Then you incubate it with phages, which process these oligonucleotides, which are biogenylated, and they display it on their surface and to which the DNA binding protein, which is specific to the oligo, that is able to bind. So these are other strategies as well. So you can read about it in a lot of detail. This is EMSA, like I talked about. This is southwestern blotting, which involves a modified Western blot using labeled oligos instead of antibodies, which you normally use during immunoblotting, because your objective is to detect DNA and you cannot detect DNA using any antibody for that matter. So that is the method. Then the Y1A or the yeast one hybrid assay, like I mentioned, which is for identifying and analyzing proteins binding to a DNA sequence of our interest. And they actually cloned the gene encoding OLF1 or olfactory neuron specific transcription factor 1. The one hybrid assay system is something we've already discussed, and the fudge display method is another one. This is PLA or for protein proximity ligation assay. Next set involves in vivo techniques, which I will not get into today, but basically it involves common techniques like chip, which is something I'll explain in the next class, native chip. X chip is also another modification. You have fast chip, carrier chip, matrix chip, chip on chip, which combines chip, which combines chromatin immunoprecipitation with microarray. Uh, then you have dip chip, which involves the modification of chip on dip is for DNA immunoprecipitation. I think if I'm not wrong. Then you have chip sequencing, the high throughput method, which is what we'll come to, and other chip variations. I'm also not very familiar with all of them, but we'll certainly learn about it. Then come your 3C and high C assays, which are chromosome confirmation capture and chip loop assay, which are more focused on the architecture of the chromatin and the 3D structure of the chromatin, regulating your gene transcription and how that can get disrupted under certain circumstances and as a response to certain kind of stimuli. So these are the different ideas. What we can certainly look at today are the in silico tools for identification of DNA protein interactions, the computational and the in silico approaches to identify DNA protein interactions are an important aspect of these. Diverse computational tools are freely available, which are used to predict DNA protein interactions, most of which are aimed at predicting the transcription factor based gene regulation. So the first one, Transpac, or otherwise also Jaspar. I'm not sure Jaspar was here, but no, it's not there. But Transpac is a tool which is utilized is a knowledge base which is utilized by Jaspar, which contains the eukaryotic gene regulation data from a number of different eukaryotic organisms like yeast to humans. It has data from transcription factors, their experimentally proven binding sites, regulated genes, and gives a good prediction tool for the same. One of the features is that it gives a score to show what is the confidence with which DNA binding activity can be annotated to a particular sequence, and it is utilized. It's sort of maintained as a relational database from which public releases are made available via the internet, making it very easily accessible. Many tools utilize Transfact, so you can go ahead and look at that. Then you have data binding protein server, you have Displar, fly factor survey, you have yeast tract, mapper, ZVP, which is ZIFIBI, which is zinc finger binding site database. Then you have bioprospector, which helps in identifying regulatory sequence motifs in the cis region of target sequence by examining it with the same gene expression pattern group, and the significance of each motif can be estimated using the Monte Carlo method. Then you have Bindin, which helps you to predict the DNA and RNA binding sites with help of support vector machines or SVMs, which are prepared using 3 sequence features like side chain PK values, hydrophobicity index, molecular mass of an amino acid. Helps to identify the function of binding proteins. Then you have Bindin Plus, which uses the protein sequence features from binding to identify binding sequences in the binding sites in the sequences, also takes the support of SVMs, and they have better performance, sensitivity, and specificity in comparison to the previous version. Then you have DB-Bind, you have PBs, Flow90, DB-PAB-B, DNA-PROT. DNA-PROT is also very commonly used, which helps in identifying the DNA binding proteins from the protein sequence, which has considerably good accuracy in distinguishing the DNA binding protein and the non-DNA binding proteins by characteristically recognizing specific DNA side chains. The random forest method is used to identify the DNA binding proteins. So anyway, rest we are not going to explore right now. Biophysical techniques, etc., is beyond the idea of this course, and I do not want to deflect from it. But just for your information, I'll just show the figure. You can do FRET, which is very commonly used, surface plasma resonance, which involves use of a sensogram. So these are biophysical methods. If that is something you are interested in, you can certainly look at it, but it is not particularly within the purview of this course. So we will stay away from that for now. And instead, I will wrap up today's class with just a quick reminder of everything we have covered till now. And the focus that we're going to assume from this point onwards. So in the next class, in class 16, we will be just studying the in vivo DNA protein interactions, focusing mainly on 1 and 2 techniques, ChIP-seq, ATAC-seq, chip, the in vivo chip, normal chip, and its variations, because that in itself is quite an elaborate method. And then we'll also look at a variation of IP or immunoprecipitation, which is either DNA independent or DNA independent, and what can be done in those circumstances, and how this is actually converted into high throughput method, which is by introduction to chip sequencing, which is what I'll also cover in the next class. Then in that will be class 16 and 17, more or less, will cover that. And then we'll have only a few more classes left, I think 6 or 7 classes left, where we'll do ATAC-seq, Hi-seq, 3C, these methods, how genomic and epigenomic biomarkers are utilized, and what is the eventual goal of integrating multi-omics. So that we'll wrap up pretty soon. So yeah, that is going to be the focus. And just to get an idea for the class, so since I've just opened up the calendar for your reference, since today is the 17th, and we had to miss a couple of classes because of some other commitments that I unfortunately had to take care of in the very last moment. Our next class is going to be on Friday, 20th, 24th, 27th, 3rd. Then again, we'll not have this particular class. So this is class 15 today. So then you have 16, 17, 18th, 19th, then 20th, 21st, 22nd, 23rd, 24th, and 25th. So by end of March, we'll be done with everything related to this course. That will include another exam for easier, better understanding, and to just, carry out a little bit of an evaluation of our current status. And yeah, that's going to be pretty much it. So thank you so much for being a part, and for attending classes on time, and for just being supportive along the way. So thank you. I am going to end the class for today. It has been a short class, but I think I didn't want to merge both of these sessions together, in vivo and in vitro, so I decided to keep it separate. But we can begin our discussion on that in the next class. So thank you so much. Have a good night. Bye.